So sometimes it's very, very important to go and actually talk to an expert. And with toxicology, I think this is really one of those cases. And David Snowden, who I've had quite a number of interactions with over the years, a toxicologist, said that there's two maxims here, two rules of thumb in toxicology. A little knowledge can be a very dangerous thing. And secondly, you can learn all you need to learn about toxicology in only 2 lessons. Each one takes 10 years. So you can go through the drill here of what I've talked about, and you can pose the limits by calculations. But I think it's much more worthwhile to talk with the toxicologist, talk with the regulatory people, and then you go to the analysts and see what you really do need for assays. Strongly recommend it. So considerations for process optimization. Quality by design is the current thrust of validation, and it is being accepted much more by the FDA than it is by the European regulatory counterparts. But it is important, and I think this is really not only the wave of the present but of the future, and we really need to consider that. QBD can really save time when it comes to, and increase the flexibility when it comes to manufacturing. It just makes good sense. Process validation really begins in the lab before the process introduction. You have to figure out what are the most important things to do and at what stage. Process validation is not just running 3 runs on a big scale in your manufacturing plant. There's more to it than that these days. DOE can be extremely helpful for optimizing your processes. And I wanted to bring out just a few examples of that. Classically, people have used OFAT or OVAT analysis, one factor at a time, changing. And that's it's a step-by-step approach, and your experiments are developed based on the results of your past experiments, one factor at a time. And you can get to where you want to go. We've done it for years, but DOE, design of experiments, are another ways to do this. And in many cases, are going to be much more effective. With DOE, you establish the order of the experiments that are going to be run before you actually start running the first. So let's assume here that you have 3 different variables. And so I haven't put the yield here, but you can have the yield labeled for each vertex of the cube, in this particular case, for 3 factors. And if you did a full factorial experiment, you'd have DOE, you'd have 8 experiments. If you did a half factorial experiment, changing 2 variables every time, you'd have 4 experiments if you had only 3 factors that you were considering, high-low. Okay, this is actually quite powerful. It can help you identify the areas for good prospecting. What areas, what parameters here do you want to investigate? Do you want to change? Which parameters actually interact? And in a half factorial DOE, you can see what are the richest places to go for further processing. So if you did half factorial, you've converted it from 8 to 4 experiments. Perhaps you run some replicates in the middle here to identify the reproducibility of your assays, of your experimentation. In which case then you wind up with 6 or with 3 replicates. You wind up with 7 experiments. How much better is that than a full factorial? Well, you can learn quite a bit more by going through the half factorial, because with replicates in the middle, because with a half factorial, you don't have any information about what's in the middle. 2 points. 2 points make a straight line, not a curve. So you need more data. And by going through a half factorial experiment with some replicates in the center, you gain information about the curvature. So if you go to a more sophisticated design, you might have what I've drawn up here, which would be actually a central composite design. And this is something that is higher level, but it can generate a three-dimensional response surface to get the job done. So again, what's the benefit of DOE? It's, say that you're going to run a half factorial experiment. You can see that as you increase the number of factors, you dramatically increase the number of experiments that are required relative to the goal. So the more complicated your system, the more factors you have to change, DOE becomes more and more helpful. To try to drive this home, I put up one example here. Pleuromutilin was being converted, natural product, over to the tricycle shown here. Now the difference is that the cyclopentene ring has migrated, if you will, to this. So what is going on is that the folks at GSK wanted to change the substituents put on here. Pleuromutilin tends to rearrange under acidic and hydrous conditions to this type of skeleton. And then under acidic aqueous conditions, this skeleton goes back. So by doing it this way, they have actually protected the alcohol group as a ketone. Then they lop off the acyl group and rederivatize it. So that's the thrust behind what seems to be a relatively curious transformation. And by going through the DOE studies, you can see that they optimize conditions pretty much to what I've shown up here. This is the response curve. You can see that there are some interactions. You can't really tell from here, but there is you want to have your optimization carried out someplace in here so that your processing is rugged. So that with a small deviation from the optimal condition, you will get very small changes in your processing and in the quality. So you want to identify the sweet spot for optimization. Now, having said that, the optimal condition here that I put up is 25 degrees. They did subsequent optimization here after the DOE studies and found, hey, 35 degrees works just fine. DOE is a tool. It's another tool that we have to try to get to where we want to go. And it's just an approximation of reality, and we need to verify it and potentially take it on to the next level and see if there are better conditions. I want to end by making kind of a plea, I guess. We run through all this wonderful chemistry in order to get from A to Z. And we want to do so in a very time-efficient manner. However, there's a lot to be gained by giving the researcher enough time to explore. And so I have some examples here to just try to drive that home. This is from Lilly a number of years ago. Now, I've had a number of people say to me, well, if you have an amino alcohol and you're going to react it with mesyl chloride, the amin's going to be the reactive site. Not true. And this is an example from Lilly, and what they found is that the alcohol got mesylated first for this particular starting material. And this was what they wanted because it generated the aziridine. So everything worked out really well. They were not able to isolate the mesylate, nor could they isolate the sulfonamide. It just went to what they wanted, not what they called it a serendipitous award for them. Now, what they indicated here is that this particular reactivity is due to the fact that they have a branched alkyl group on the nitrogen. So it depends. Another example here of how important it is to have a little bit of leeway to explore and to pay attention. A number of years ago, Pfizer folks got a green chemistry award for this particular route to get to sertraline. This is a sertraline intermediate. The mandelic acid salt is not pharmacologically approvable. The HCl salt is what is the API. But their yield went up, almost doubled by doing this processing. And if you take a look at it critically, what you see is that everything is driven by the solvent choice, ethanol or methanol. The intermediate crystallizes. Boom. The reaction, this is an equilibrium reaction. You produce water. Water should drive it back in that direction. But just by virtue of the fact that the imine crystallizes out means that they don't have to use any other reagents to dehydrate the system, such as titanium tetrachloride, which is what they probably used. And that's how the whole thing is driven, by just noticing, hey, it crystallizes under these conditions. Sweet. But let's take the sertraline one step further here. This I found quite curious. Alpha tetrol reacts with dichlorobenzene, neat, 8 equivalents in the presence of aluminum chloride to generate this adduct. This is an adduct here in 80% yield. Now, I don't know about you guys, but certainly in my knowledge of Friedel-Crafts reactions from Morrison and Boyd and all those years ago, I would not have expected this. And this is what the Hungarian group has reported. And they have a procedure there in order to generate this material. Now, I suspect, and they described taking this racemic material and converting it over to sertraline. But the folks from Pfizer, Joel Hawkins, talked about using SMB to separate these two enantiomers. And once you use SMB or SFC, whatever you are dealing with, you can take the right enantiomer and convert this on through to sertraline. So again, all of this is predicated by this, to me, unusual product from a Friedel-Crafts type of condition. So process introduction, just to my perspective anyway, few scale-ups are flawless. It happens, but what we can do is to try to anticipate the problems and avoid them. That's really our job. Often the problems on scale-up arise from controlling the temperature, controlling mixing, and last of all, contamination. It's so important to make sure that the reactors have been cleaned, and then secondly, have all the cleaning fluids been removed from the reactor. If you have water left in the recycle loop that's used for sampling, and you're running a reaction with thionyl chloride, you are probably going to be hurting. So contamination is a big deal. Priorities change. We expect that processes will change as time goes on. They may change because there's a particular impurity that's been produced and your analyst just uncovered it with a new HPLC system. They may change because now you have to get it really cost effective. So things change. We expect processes to change too. So my summary, Rapid commercialization requires efficient process development and partnership. Organic chemists, chemies, analysts, toxicologists, the regulatory folks, others here which you could probably list based on your own experiences, but it's something that we really do need the partnership. As process chemists and engineers, we can anticipate a lot of the difficulties, and it's our job to do so, but we really need to call in the whole group to get the things done. So that's where I'd prefer to leave things, and I'll say thank you very much for your kind attention and coming out here today. If there are any questions, I'd be happy to address them. Thank you.
Neal, excellent talks. Really enjoyed it and actually represented a lot of those remarks. So the genotoxic, I guess the potential and mutagenic material guidance that you gave for the duration of use, is that an official guidance, or is that sort of proposed or being proposed, or?
F4, it's the final. It's the final guidance, Sharon.
Hey, thanks for coming. And one thing I would ask, if you think of any other good speakers that we could bring in for another seminar, maybe in 6 months, let me know, and let's bring someone else in here. Thanks a lot.